Hey everyone, and welcome to the aerobic metabolism lecture that's going to cover the electron transport chain, okay, or ETC as we'll call it. A little overview as we get into things. So, the electron transport chain, or ETC, is important because it uses those pairs of electrons and hydrogen protons or atoms to create a lot of energy. And throughout the process of the transport chain, it does it through two different means. Okay, one of the main ways is, sorry, just to clarify, they happen at the same time. So it's like a simultaneous, kind of continuous process between these two components of the electron transport chain that generate ATP. So one of them is pairs of electrons. As you'll see, they'll get passed between different holders in this process called cytochromes. You can think of cytochromes as protein structures that sit within the cell membrane of mitochondria. And we'll give you a more specific location shortly. But as these electrons get passed, what happens is at 3 points along the chain, there's enough energy transferred or converted where ADP can capture some of that and get phosphorylized. So it adds a phosphate group and becomes ATP. Okay, and the way this works is these electrons get passed from an inner membrane to an outer membrane of a mitochondrial organelle in the cell. So to think back to maybe some earlier bio, or maybe this is a bit of a new image that you can put in your head, but you can think of a mitochondria as like a sack or a jelly bean, and then within that bag or jelly bean, there's actually a second bag that kind of looks like, I guess, intestines a little bit, but that's right in the middle. So the inner membrane is encapsulated by the outer membrane. And so you can have something from that inner compartment still get outside of that inside bag, and it'll still be within the mitochondria. I'll show you some images in a bit, and it'll hopefully make more sense. But what happens with these, so we get these electrons passing along, and what they do is they cause these pumps to pull or move these hydrogen protons from one side of the membrane to another, and creates this gradient. Okay, so from the inner part of the mitochondria to the outer membrane. And what's important about this is it creates something called a concentration gradient. This is a really big thing in biology just in general, but the membrane itself can't have the hydrogen protons pass through. So if our cells keep pumping all these protons into this holding area, they can't move anywhere. Now, based off of how chemicals like to act, when there's a gradient of something of high concentration and a concentration of something low, inherently equilibrium is wanted to be established. So what I mean by that is the protons in the high concentration area want to move to the low concentration area, but can't because of that membrane. Well, they can't yet. Okay, so this is actually a big way how renewable resources right now for energy are hoped to be kind of utilized and leveraged in the future. So, for example, some strategies that are being developed are using, say, solar energy during the day to pump water up into a reservoir at the top of a hill during the day when there's lots of light. But then that unused energy that has been used to pump up the water in the evening, since there's no sun, what they'll do is they'll slowly release some of the water from this holding reservoir at the top of the hill. And as we know, water will naturally flow downhill. And so as that water comes down and it turns essentially like a paddle wheel or like a turbine, as the water comes down, it'll generate electricity in the evening that way. And then during the day, it gets pumped back up and then flows down in the evening. And so I use that as a specific example because the electrons in the ETC do exactly that. They pump hydrogen protons up across this gradient, and then we'll see this protein molecule or gate with a turbine in the membrane of the mitochondria does just that. It allows hydrogens to flow down through this protein structure, and essentially, quite literally, turn a component of the ATP synthase, which is what the molecule is called, that allows this to happen, to create ATP. So it's really, really cool. Again, we'll get to some images shortly to help visualize this and see what's going on. But yeah, so ATP synthase creates a lot of ATP because so many hydrogen protons are pumped across the electron transport chain as the electrons move across the different protein structures in the mitochondrial membrane. Okay. To wrap it up at the very end, oxygen accepts those electrons as well as hydrogen protons to make water. So we get water as a byproduct of our oxidative respiration or phosphorylation. And just as a last little note here, we'll talk about numbers of ATP produced per metabolic substrate in a following video, but we'll talk a little bit about what is produced at the end of the electron transport chain here. Okay. So again, as promised, here's a schematic to help us kind of digest, reimage. I'll repeat so you can hear this information again, what we just covered. But here's our diagram of the mitochondria. And as you can see, we have our outer membrane and this little inner membrane, which is not permeable to hydrogen ions. And here's our electron transport chain and our ATP synthase, or as they also call it, respiratory assembly. Okay, it's the same thing. So how does this work? Let's hear it again. Let's go over it again. So what's important to notice is that these actual kind of structures that rest within this inner membrane of from the outer shell, so this outside shell, and the inner membrane, so this component in here, the inside squiggle, the intestine looking part, those are the electron transport chain proteins. And as you can see here, this is our electron that bounces across these cytochromes, right, to pass along. Every time that happens, that energy that gets released or utilized pumps from here in the inner membrane to the outer membrane hydrogen protons. So the first pump pumps 4, and again that's collected from, as you can see here, NAD or NADH, and then becomes replenished as NAD in order to go get more hydrogen elsewhere off of, say, Krebs cycle. So 4 hydrogens go up into this storage area between the outer and inner membrane of the mitochondria. Same thing happens again with the second cytochrome or hydrogen pump. The third one, however, only pumps up 2. And the reason for that is the other 2 that would normally, I guess, get pumped through, it's accepted by oxygen, along with the electrons that had been jumping along here. They all get accepted here as water, and then either used in parts of the Krebs cycle, used in the cell. Water is kind of ubiquitous and can get used in a number of different ways. But here we have it at the end of our electron transport chain. And so again, this is one primary way, as we can see, happening simultaneously with the second way the electron transport chain makes energy, where ATP is. The electrons are one component. The second component is the hydrogen gradient that gets pumped up into this membrane between the outer and inner membranes of the mitochondria. Now, I'm sure you're wondering, you have this question in your heads, I'm sure. Okay, we've spoke about the electrons. We know we have this concentration gradient with the hydrogens. How does this hydrogen concentration actually make ATP? Because so far, we see that over here, but what's going on with this respiratory assembly, or ATP synthase? Well, let's zoom in a little bit. We'll kind of get rid of the larger schematic of the mitochondria. And what we can see here is, again, we have these hydrogens pumped up. So that's 4, 8, 10. And again, just highlighted for use as oxygen for aerobic respiration as a recipient or final receiver of the electrons and protons. Hydrogen protons at the end here. It turns into water. Okay. We've covered that redundantly now. Okay. Great. So this respiratory assembly, what's going on there? Well, just like the renewable solar power water storage complex that I was explaining earlier, where water gets pumped up the hill, flows down at night, we have the same thing with a rotor here. And I think what helps is to actually view ATP synthase, or again, remember it's got two names, the respiratory assembly, in its actual form. So this schematic, I think, is misleading. It looks just like another channel or cytochrome within the ETC, and it's really not. It's its own thing, ATP synthase. So I'm just going to play this in the background. It's posted in Blackboard for you to be able to look up and view in its entirety for you. But what I'm hoping is demonstrated here for you is you see this high hydrogen proton concentration up here. So get yourself situated with this schematic, a detailed schematic of the protein structure of ATP synthase here. Up above this part, most superior, is where the proton gradient is high. Down here is where it's low. So again, in the video, as you can see, this component here is like an axle or a rotor. So, and as hydrogen protons flow down through it, it quite literally turns these protein structures to rotate. And as it does it, it opens up these little pockets in this lower component that allows ADP and phosphate to enter and become phosphorylated and become ATP. Pretty cool, hey? So again, the exact number of ATP that's produced we'll cover a little bit later, but the main thing that I wanted you to focus on is, again, this acts like an actual mechanical rotor that as these protons from up between the outer and inner membrane of the mitochondria flow through these. Again, so this is closing up the 3D image for you. So it was transected before, now it's not. So as it flows through all these rotors, we generate a lot of ATP just through this concentration gradient that occurs. Okay, I think this stuff is wild. I like the fact that we can visually and through different measurements in biology have visualized this through different methods is unreal. The fact that we can essentially track that this happens, unreal. And the fact that our bodies have this capacity within our cells that are just constantly operating is like right now it's keeping you alive, quite literally. Absolutely amazing. This is the coolest stuff I think that's possible. It's out there. So anyway, yes, so we got our ATP synthase. This is a simplified version, okay? So just hopefully this illustrates the difference between the way that we often view these processes as far as biochemistry and what it actually looks like and is going on is quite dramatically different, of course. But just to keep that in perspective, again, hopefully here it just lays it out a little cleaner for you. You can see 1, 2, 3 ATP being pulled off the electron transport chain itself quite directly. Down over here, we have our ATP synthase, kind of as a second component. We're going to talk about how much ATP that generates in the next section. But again, just purely from those electrons moving 3 times between the cytochromes, we have 3 ATP produced from the hydrogens from NADH and FADH2. Okay, that's it for the ETC. Cheers.